So when I started my lab in 1982, I started working on polio virus, and for many years we worked on it, but as you may know, it's about to be eradicated, so we've had to turn to other viruses. A few years ago, we got very excited about Zika virus because Zika virus was shown to be able to infect the brain, and that's something that we had studied with polio for many years. So we worked on Zika, and then we got excited about a virus called Enterovirus D68, which is related to polio and causes a respiratory illness in children, and in a small fraction of them, they get paralyzed. So it's very much like the disease that polio virus causes. And so we're starting a research program to work on that virus. We want to understand how it causes paralysis, how it gets from the respiratory tract to the brain, why certain kids might be more susceptible to infection by others, and of course develop ways to detect and prevent infection. So we're really excited about that particular virus right now. It's great that it's about to be eradicated, and the fact that we can't work on it anymore shouldn't really matter. A lot of scientists have had to stop working on the virus because it's about to be eradicated, but that's the ultimate outcome of research, right? You get rid of a human pestilence. So I think it's wonderful, and as I've said, we've switched to other viruses, so it's not a problem. If every scourge could be eradicated, it would be great. As you know, smallpox was one of the viruses that we have eradicated. Polio will be next. Measles is another one that we could eradicate if we could convince people to be vaccinated against it and so forth. But I think it's the ultimate outcome of your work that a virus could be eradicated. Polio virus is ingested with fecally contaminated food, dirty hands. It goes into your intestines and it multiplies, and then it's shed in feces, and that's how it spreads to other kids. And in a small fraction of infected kids, 1 in 100, the virus gets into the brain and spinal cord and the kids get paralyzed. That's why we developed vaccines, because American President Franklin Roosevelt got polio and he said, we got to stop this. So we were studying over the years how the virus got from the intestine to the brain and spinal cord, how it got in, what it did once it got in there, how it destroyed neurons to cause paralysis. We actually developed a mouse that we could use to study this, a mouse model for paralytic disease. So we'd like to do similar things with this Enterovirus D68, except in this case, it's going to infect the respiratory tract. So we'll say, can we make a mouse that duplicates all of the same things that happen in people? Respiratory tract infection, spread to the brain and spinal cord, entry, and then destruction of nerve cells to get paralysis. Because we don't know anything about how that happens in people, and the best way to do that is to make a mouse model. First picornavirus ever discovered was polio virus, and that was in 1906. And then there were others discovered in the 50s and 60s. When I was a postdoc, I think there were 4 different picornaviruses known. Nowadays, there are thousands. Wherever people look, they find picornaviruses, whether it's in mammals or birds or reptiles or even invertebrates or insects. You find picornaviruses everywhere. They infect almost everything on the planet, and certainly in humans, they cause millions and millions of infections a year. And the only ones we really have studied very much are the polio viruses and maybe the rhinoviruses. So there are a lot of other picornaviruses that infect people that aren't studied, and we need to study that. And Enterovirus 68 is a great example of why we should be doing that, because 4 or 5 years ago, all of a sudden out of nowhere, it started causing epidemics. We'd known about it since the 60s. All of a sudden it started causing epidemics, and we were totally unprepared for that. So now people are catching up, trying to understand how the virus is doing all these things. So the main reason is that there are lots of them out there infecting many people and many non-human living things on the planet. So we need to know how all that is interrelated. So innate immunity is really important, because if you make mice that lack innate immunity, they get overwhelming picornavirus infections. And we have done this, and other groups have done this as well. So when the virus first infects an animal, whether it's a human or a mouse, it triggers innate immunity, the production of interferons, and in most cases, those can reduce virus replication and prevent subsequent infection. And people that get serious picornavirus infections most likely have mutations in genes involved in innate immunity that make them more susceptible to infection. There's some very interesting examples. For example, children who get recurrent respiratory infections by rhinoviruses, they have defects in their innate immune system. So innate immunity is essential for preventing infections in most of us, and so we need to understand that better, and many, many laboratories are working on that. The main problem is that they're all pretty quick infections. We call these acute infections. They infect you within a few days, you have symptoms, and then the infection's resolved. And so we don't have time to treat. By the time you are sick, you would have to go to a doctor, get diagnosed, get your prescription, go to the drugstore, get it. By then, it's too late. And so what's going to be needed is to have rapid diagnostics, where you could go to a physician office, they could do a test right there and say, you have a rhinovirus infection, here's the prescription, and go get it. But we don't have rapid diagnostics. Therefore, companies are not making antivirals. So it's a chicken-egg problem. Who's going to be first? Now, rapid diagnostics is an advancing field. Dare I say rapidly advancing field. And so people are doing this for other viruses, and it will just be a matter of time where it gets applied to picornaviruses, and then we will have antivirals. Very few have been tested in the past, but they haven't worked very well, and resistance emerges rapidly. So we're going to need more than one. Like the situation for treating HIV/AIDS, we need 3 drugs. And so for picornas, we're going to need 3 different drugs to treat the patients with that so that we won't get resistance. I think it's a good idea. If you understood how polio got from the blood into the brain, maybe you could apply that to getting drugs in. But no one really has looked at that in the field. But for polio, I don't think we'll be able to figure that out because it's about to be eradicated. And when it's eradicated, we can't really work on it any longer. But there are certainly other viruses that do that. And I think that is a really good approach to understanding better drug delivery. Yeah. I've been very lucky my entire career to be incredibly curious and passionate about our viruses. And I would say to anyone who's thinking about doing research of any kind, you have to be passionate and you have to be curious, and everything else will follow.